Some diseases can begin with a tiny change in DNA. That small change can sit inside a gene, like a typo in an instruction. CRISPR is a way to find a chosen DNA address and edit it. First, we need to zoom from the whole cell down to the code. The cell contains a nucleus, and the nucleus contains DNA. A gene is a useful section of DNA that gives the cell instructions.
The question is, how can a tool find one small section inside so much code?
We move from the cell to the nucleus, then to the DNA itself. Inside the nucleus, DNA is packed and folded, but it still follows a sequence. CRISPR does not search by eyesight. It searches by molecular matching. The target is a short sequence, not the whole chromosome.
That is why the guide RNA matters so much.
It gives Cas9 a specific address in the genetic code.
Once we know the address, the editing story can begin.
DNA is written with 4 letters: A, T, C, and G. Different orders of these letters can change what a cell builds or controls. A gene is one meaningful instruction inside that long DNA text.
If the instruction is damaged, the outcome may change.
CRISPR gives scientists a programmable way to point at one instruction. Before it became a lab tool, CRISPR was part of bacterial defense. Bacteria can store small fragments of viral DNA as molecular memory. When a similar invader appears again, the system can recognize it.
That memory idea was adapted into a programmable targeting tool.
The lab version uses a guide RNA and a cutting protein, such as Cas9.
Guide RNA is the address label.
Its letters are designed to match the DNA sequence we want to target. When the guide lines up with the DNA, base pairing helps confirm the location. A good guide is specific enough to reduce wrong matches.
Now Cas9 can be brought to that location.
Cas9 is the molecular cutter that travels with the guide RNA. It opens the DNA and checks whether the guide really matches. Many Cas9 systems also need a short nearby PAM signal. If the guide and the landing signal are right, Cas9 locks in place.
Now the target is selected and the cut can happen.
At the target site, Cas9 cuts both strands of DNA.
This creates a double-strand break, a serious signal for the cell.
CRISPR does not decide the final repair by itself. The cell's own repair machinery responds to the break.
That repair response is what creates the edit.
After the cut, cells can repair the break in different ways. One path is NHOJ, a quick repair that often adds or removes tiny pieces. That can disrupt a gene and turn its function off. Another path is HDR, which can copy a supplied repair template. HDR can replace a faulty section with a corrected version. So the same cut can lead to different editing outcomes. A knockout edit is useful when researchers want to disable a gene. A correction edit tries to restore or replace a specific sequence. The difference is not only the cutter, it is the repair pathway and template.
That is why experimental design matters. Gene editing is a process, not just a pair of scissors.
CRISPR is used widely in research to test what genes do. It is also being studied for medical therapies and diagnostics. In agriculture, gene editing can help study traits and resilience. But every application needs careful delivery and testing. The tool is powerful because the target can be programmed.
Powerful tools also need strong safety checks.
A guide RNA can sometimes match a similar but wrong location. Delivery into the right cells can be difficult. The repair outcome is not always the one you want. That is why off-target screening and long-term testing are important. The goal is precise editing with responsible control.
The whole idea can be remembered in 3 words: find, cut, repair.
Guide RNA finds the address. Cas9 cuts the target DNA. The cell repairs the break, sometimes disrupting and sometimes correcting the gene. CRISPR is not a magic wand. It is a programmable editing system. When used carefully, it lets biology be studied and changed with new precision. Find the target, cut the DNA, and guide the repair.